name is Dr. Jacqueline Miller, and I am the Senior Vice President and Therapeutic Area Head of Infectious Diseases at Moderna. I'm pleased to share with you today some of the details of our clinical development program and our key immunogenicity and efficacy results. Before moving to our clinical program, I would like to review our key non-clinical results. We generated an extensive non-clinical data package in 3 different animal models, including non-human primates, or NHPs. Our data demonstrate that mRNA 1273 induces humoral and cellular immunity, including memory B cells in vaccinated animals. We also challenged these animals with SARS-CoV-2 virus and found that the vaccine could fully protect animals at subtherapeutic doses. No evidence of vaccine-associated enhanced disease. We have recently completed our developmental and reproductive toxicology study, indicated no safety concerns. Development of mRNA 1273 has been accelerated given the COVID-19 pandemic. Nonetheless, a full development program, including phase 1, 2, and 3 studies, has been executed. Study 101 was our phase 1 dose-ranging safety and immunogenicity study conducted across 3 age strata, 18 to 55, 56 to 70, and over 71 years of age. Study 201 was a phase 2 safety and immunogenicity study. The primary focus of our presentation will be the phase 3 COVID-19 Efficacy and Safety Study, or 301, as it enrolled over 30,000 participants, approximately 15,000 of whom received mRNA 1273. Study 301 generated the vast majority of safety and all of the efficacy. So let's begin with Study 101. This slide summarizes the neutralizing antibodies induced by 100 micrograms of mRNA 1273 across 3 age strata. Shaded area represents a panel of titers, a range of titers from a panel of convalescent sera taken from individuals recovering from COVID-19 disease. Served as the clinical benchmark to compare immunogenicity between the doses and the samples were 23 to 54 days after diagnosis. Neutralizing antibodies were induced in all participants by day 36, or 1 week, dose 2. GMTs were comparable across the 3 age strata, including participants in the older age strata, and persisted until day 119. Now, let's discuss the T-cell immunity evaluated in Study 101. CD4 T-cells were further evaluated for Th1 and Th2 phenotypes, since T-cells are thought to be associated with enhanced disease. The top panel of this slide represents the Th1 phenotype, and the bottom panel is the Th2. Th1 dominant CD4 T-cells are induced by day 43 across age strata. Minimal detection of the Th2 phenotype. This analysis showed no evidence of enhancement. I'd now like to present the immunogenicity results for the 100 microgram and placebo groups in the Study 201. Dark blue bars represent the 100 microgram dose, and the gray bars represent placebo. By day 43, there was more than a 50-fold increase in geometric mean titers in the vaccine group. And in the placebo group, GMTs remained below the level of quantitation. So in summary, our phase 1 and 2 studies showed the induction of neutralizing antibody titers in all participants by 1 week following the second dose. GMTs were observed to be higher than those of a panel of convalescent sera, and neutralizing antibodies persisted for 3 months after the second dose across all 3 age strata. Th1 dominant CD4 T-cell response was also observed across age strata, was consistent with our findings in animal models. So now, let's look at the efficacy data from Study 301. Study 301 was designed to evaluate the efficacy, safety, and immunogenicity of mRNA 1273 compared to placebo in adults at least 18 years of age who were at risk for COVID-19. 30,420 participants were randomized 1-to-1 to receive 2 doses, vaccine or placebo. Participants received the first dose on day 1 and the second dose 1 month later on day 29. Participants have been monitored for efficacy, immunogenicity, and safety endpoints throughout the study. Immunogenicity endpoints include the measure of binding and neutralizing antibodies at the indicated time points. These immunogenicity samples will also be used to assess for asymptomatic seroconversion to non-vaccine antigens. These data were not available for the emergency use submission and will not be discussed for today. Efficacy surveillance occurred throughout the study. Once diagnosed with COVID-19, participants underwent daily telemedicine visits to ensure close medical follow-up. Participants were also given pulse oximeters to measure their oxygen saturation daily. Study 301, primary efficacy objective, was based on COVID-19 cases that occurred in SARS-CoV-2 seronegative participants. To demonstrate success, the lower limit of the 95% confidence interval for vaccine efficacy had to be greater than 30%. Secondary endpoints for vaccine efficacy include the evaluation of efficacy against severe disease and death, COVID-19 using the CDC case definition, and COVID-19 cases occurring after the first dose. There was also a secondary objective to evaluate asymptomatic SARS-CoV-2 infection, but the results are not yet available. Please let me review the case definition for COVID-19, severe COVID-19 disease. Primary efficacy endpoint: symptomatic, adjudicated COVID-19 disease that occurred at least 14 days after dose 2. To be considered a case of COVID-19, study participant had to have experienced at least 2 systemic symptoms or at least 1 respiratory sign or symptom or clinical or radiographical evidence of pneumonia and confirmed SARS-CoV-2 infection from at least 1 nasopharyngeal. Study 301 also analyzed efficacy against severe COVID-19. Severe cases had to meet all criteria for the primary endpoint and have at least one of the 4 following criteria: severe systemic illness or respiratory failure, acute respiratory distress syndrome or evidence of shock, or significant acute organ dysfunction or admission to an ICU or death. To ensure adequate safety monitoring and to enable the interim efficacy analyses, this study has been monitored by a Data and Safety Monitoring Board, or DSMB. DSMB was chartered and convened by the National Institutes of Health and is completely independent from the company. In addition, an independent efficacy endpoint adjudication committee was assembled to determine if the case definitions for COVID-19 and severe COVID-19 were met. The committee has adjudicated all cases for the primary efficacy endpoint and continues to adjudicate cases as they accrue and will ultimately adjudicate all COVID-19 cases reported. 30,420 participants were randomized in Study 301, including 15,010 subjects to each group. The full analysis set includes 15,181 participants who received at least 1 dose of mRNA 1273. The modified intent-to-treat population includes participants who had no evidence of infection prior to receiving their first dose of study vaccine or placebo. For protocol population, predefined for the primary efficacy analysis. It includes participants in the MITT who received both planned doses and had no major protocol deviations. More than 92% of participants vaccinated in both treatment groups are part of this population. Now let's return to the efficacy results. Enrollment was stratified to ensure that we studied participants most at risk for COVID-19. We pre-specified that at least 25% of our study population would include participants over 65 years of age or subjects between 18 and 65 with comorbid medical conditions. We were successful and enrolled a total of 42% of the study population in these two categories. Let's review the study demography by gender and age. Approximately equal proportions of males and females participated, and the mean age was 51, the range of 18 to 95 years. 25% of the study population was over 65 years of age, and half of those individuals were over 70. Age and gender distribution were well balanced between. This trial included approximately 10% African Americans, 5% Asian American, and 21% of participants who identified as being Hispanic. This is a breakdown of the comorbid conditions reported in the study. 23% of participants overall reported at least one pre-existing condition. This included 9% with diabetes mellitus, 7% with severe obesity, 5% each with significant cardiac disease or chronic lung disease. A specific inclusion criterion was that participants had to be at increased risk for COVID-19. Overall, 25% of our study participants are healthcare providers, and a substantial proportion of the remaining subjects meet the definition for essential workers. Taken together, the participants depicted on this table represent more than 50% of our study population. So here are the numbers of COVID-19 cases contributing to the primary endpoint by demographic subgroups. 33 cases occurred in the elderly, including 10 of the severe cases. 42 cases occurred in people from communities of color that have been disproportionately impacted by COVID-19. This slide displays the primary efficacy results for the pre-specified interim analysis. Primary efficacy hypothesis was met. Vaccine efficacy after the second dose was 94.5%, the lower limit of 86.5%. Difference between groups was statistically significant, the p-value less than 0.0001. The incidence rate in the vaccine group was 1.8 as compared to 33.4 thousand person years placebo group. This interim analysis was submitted as part of Moderna's EUA application, currently under review by the EUA. A second analysis was performed when the full pre-specified cohort of 151 cases COVID-19 had accrued and the 2-month median follow-up time point had passed. This analysis was predefined in the protocol as the primary efficacy analysis. There were 196 cases, 11 of which occurred in the vaccine group and 185 occurred in the placebo group. Vaccine efficacy was 94.1%, the lower limit of 89.3%. Difference between the groups was also statistically. Incidence rate was 3.3 in the vaccine group compared to 56.5 in the. Now, I would like to show you a forest plot of various subgroup analyses we performed on the primary endpoint, stratifying the population by age, gender, race, and risk factors. All subgroup analyses were consistent with the primary analysis, lending confidence to the generalizability of the efficacy. Also evaluated the efficacy of mRNA 1273 against severe COVID-19, the secondary objective. 30 severe cases have been adjudicated at the time of the primary efficacy analysis, and all occurred in the placebo group, resulting in a point estimate of vaccine efficacy 100%. There's also a single death due to COVID-19 reported in the placebo group. We have also evaluated efficacy according to the CD case definition, which required only one clinical symptom from an expanded list and a swab positive for SARS-CoV-2 virus. Point estimate of efficacy with this definition, 95.1%, highly consistent with the primary efficacy hypothesis. We have also investigated the efficacy against cases of COVID-19 occurred 14 days after dose 1 as a secondary objective. There were 11 cases in the vaccine group compared to 225 cases in the placebo group, for an overall estimate of vaccine efficacy 95.2%. The result is limited by the fact that not all cases are adjudicated. More than 96% of participants received their second dose, and the analysis included cases which occurred after the second dose. Nonetheless, the fact that the efficacy estimate is so consistent with the primary analysis is Kaplan-Meier curve. The cases that occurred in the modified intent-to-treat cohort since randomization are shown on this slide, supporting the secondary efficacy analysis for efficacy after the first vaccination. Based on this, we also evaluated the percentage of subjects in the modified intent-to-treat cohort according to the CDC case definition, which occurred after randomization. We'll see on the next slide. So these are all the cases reported in each group, stratified by 2-week intervals up to the second dose. Overall, prior to 14 days post-dose 2, there were 62 cases in the placebo group as compared to 8 cases in the vaccine group. Most of the cases in the vaccine group were reported in the first 2 weeks after vaccination. Taken together, these analyses suggest that protection may begin prior to dose 2, but for maximum protection, both doses should be given. Our protocol-specified analysis on the efficacy against asymptomatic infection was not available at the time of the EUA submission. However, did collect pre-dose 1 and pre-dose 2 swabs for SARS-CoV-2 virus and have performed a descriptive summary comparing the number of positive swabs as a way to estimate asymptomatic infection. Among baseline negative participants, 14 in the vaccine group and 38 in the placebo group had evidence of SARS-CoV-2 infection at the second dose without reporting symptoms. There were nearly 2/3 fewer positive swabs in the vaccine group as compared to the placebo group at the pre-dose 2 time point, suggesting the possibility for prevention of asymptomatic infection. So in conclusion, mRNA 1273 has demonstrated clear and compelling evidence of vaccine efficacy against symptomatic COVID-19 disease. Vaccine efficacy was 94.1%, the lower limit of the 95% confidence interval of 89.3%, successfully meeting the primary efficacy hypothesis and exceeding the FDA guidance for COVID-19 vaccines. At the time of the data cutoff, 30 cases of severe COVID-19 had occurred in the placebo group, and no cases had occurred in the mRNA 1273 group. Efficacy against severe disease is reassuring about the lack of enhanced disease, and participants in this trial will continue to be followed for breakthrough disease. All key secondary, sensitivity, and subgroup analyses were consistent with primary analysis, underscoring the performance of the vaccine across high-risk populations. Given this high and consistent efficacy, mRNA 1273 offers the potential to address the public health crisis of COVID-19. Thank you.